Welcome to your Introduction to Statistics Unit 6, Lesson 4. Today we're going to talk about the 4 principles of experimental design, and we're going to see how to diagram experiments. The first principle of experimental design is control. We control sources of variation other than the factors we are testing by making conditions as similar as possible for all treatment groups. The second principle is to randomize. Randomization allows us to equalize the effects of unknown or uncontrollable sources of variation. So we control what we can, and then we randomize to control for the things that we can't actively control for. It does not eliminate the effects of these sources, but it spreads them out across the treatment levels so that we can see past them. Without randomization, you do not have a valid experiment and will not be able to use the powerful method of statistics to draw conclusions from your study. You won't be able to establish a cause and effect relationship. The third principle is replication. We repeat the experiment, applying the treatments to a number of subjects. The outcome of an experiment on a single subject is an anecdote, not data. So you have to have many subjects within your experiment. And that's the kind of replication we refer to specifically here. Another kind of replication that is good is that if the experimental group is not particularly representative of an entire population of interest, you might want to replicate the entire experiment for different groups in different situations. The fourth principle is to block or blocking. Sometimes attributes of the experimental units that we're not studying and that can't be controlled may nevertheless affect the outcomes of an experiment. Like if you think gender really plays a role in the outcomes, you might block according to gender. If we group similar individuals together and then randomize within each of these blocks, we can remove much of the variability due to the difference in the blocks. Note that blocking is an important compromise between randomization and control, but unlike the first 3 principles, it is not required in an experimental design. It's often helpful to diagram the procedure of an experiment. The following diagram emphasizes the random allocation of subjects to treatment groups, the separate treatments applied to these groups, and the ultimate comparison of the results. So if you look, you start off and you describe the random allocation of however many subjects you have, and then you've got it split off into your groups. Typically you say how many people are in each group, and then you describe the treatment. And then at the end you compare, and you don't just put compare. They have it here just, to serve as a pattern. You put whatever the response variable is. Whatever you're actually going to compare, you put out here to the side. Now, when we're comparing, we're looking for a difference in the response variable, and that brings up a question. Does the difference make a difference? How large do the differences need to be to say there's a difference in the treatments? Okay, how big does the difference in the response have to be to say that the treatments actually did something significant? Differences that are larger than we get just from randomization alone, just due to chance, are called statistically significant. Both experiments and sample surveys use randomization to get unbiased data, but it's important to understand the 2 different places or ways that randomization is used, and they're done in different purposes. Sample surveys try to estimate population parameters, so the sample needs to be as representative of the population as possible. So we do random selection of the subjects from the population. Experiments try to assess the effects of treatments, and experimental units are not always drawn randomly from a population. You have to work with volunteers. So then you take those volunteers and you randomly assign them to the treatments. Often we want to compare a situation involving a specific treatment to the status quo situation. A baseline business as usual measurement is called a control group, and the experimental units to whom it is applied is called the control group. So you have a control treatment, and the group is called the control group, and that is so that you have a basis of comparison. When we know what treatment was assigned, it's difficult not to let the knowledge influence our assessment of the response, even when we try to be careful. In order to avoid that bias that might result from knowing what treatment was assigned, we use blinding. There are 2 main classes of individuals who can affect the outcome of an experiment. those who could influence the results, the subjects themselves, the treatment administrators, the technicians, and those who evaluate the results, judges, treating physicians, etc. When every individual is in either one of these classes is blinded, an experiment is said to be single-blind. When everyone in both classes is blinded, the experiment is said to be double-blind. Now, sometimes students get confused because they think if it's double-blind, well, then nobody knows who's getting what treatment. No, there's one person who typically has the key to the experiment, who knows which people are receiving what treatment. But those who are actually taking the treatment, those who are giving it to them, those who are judging the results, they don't know until the very end. So somehow it's coded, and only the keeper of the key knows. Okay, one of the ways that control groups work is that people are given a placebo. Often simply applying any treatment can induce an improvement. To separate out the effects of the treatment of interest, the one that we actually think has some sort of therapeutic effect, we can use a controlled treatment that mimics the therapeutic treatment itself. A fake treatment that looks just like the treatment being tested is called a placebo. Sometimes on TV you'll hear people talk about, well, they had this percentage more improvement than those who received a sugar pill. That's another way of saying a placebo. It's just a pill that looks like the treatment pill, but it has just sugars or something in it. Placebos are the best way to blind subjects from knowing whether they are receiving the treatment or not. The placebo effect occurs when taking the sham treatment results in a change in the response variable. This highlights both the importance of effective blinding and the importance of comparing treatments with a control. Placebo controls are so effective that you should use them as an essential tool for blinding whenever possible. It's amazing how much just believing that you're doing something that will improve your health or something else, how much of an effect that can actually have on you. The best experiments are usually randomized, comparative, double-blind, and placebo-controlled. So there's random assignment of subjects to treatments. They're comparative. There's at least two groups, at least a control group and a treatment group. In that control group, you may also have the placebo-controlled aspect of it, where the treatment is placebo. And double-blind means neither the subjects or anybody giving them the treatment knows whether which treatment they're receiving, and the people who are judging the response variable, they don't know either. Someone else is not actively involved in interacting with the subject, either giving the treatment or measuring the response. That person who's kind of further removed from this study knows who is receiving what treatment. Now, we said sometimes blocking is used. When groups of experimental units are similar, it's often a good idea to gather them together into blocks. Blocking isolates the variability due to differences between the blocks so that we can see the differences due to the treatments more clearly. When randomization occurs only within the blocks, we call the design randomized block design. So if we thought that men and women were going to react differently to a medication, you wouldn't want just by the random assignment for the majority of the men to end up in one group and the majority of the women ending up in the other group, because the response that you measure may not be due to the treatment itself. It may be measuring how men respond and how women respond. So what you would want to do is you would want to block according to gender and then randomly assign half of the men to one treatment, half of the men to the other treatment, half of the women to the first treatment, half of the women to the second treatment. That way, you and then pull the results according to treatments at the end. That way you have the same proportion of men and women in both groups. Here's a diagram. It's not with men and women. It's actually with tomato plants. And so an experiment was being done to see if a particular plant food resulted in creating juicier and tastier tomatoes. And the experiment designers, the experimenters, wanted to know if a half dose works just as well as a full dose, or so there's three different groups: the control group, the half dose group, and the full dose group. they were not able to buy all of their tomato plants from a single store. They bought them from 2 different stores. And the thought was, well, even though they're the same type of tomato plant, the different stores may have had different treatments. They may have treated the plants differently. And one store may have more robust, better plants than the other. One store may provide, tastier, juicier tomatoes just because of the way that that store treated the tomato plants before the experimenters ever got them. So what they decided to do was block according to the store where the tomato plants were purchased. So up here you see block A, and those are the 12 tomato plants purchased from store A. And then here we have block B, the 6 tomato plants from bought from store B. So what they did was they randomly assigned a third of the 12 tomato plants from block A, from store A, to each of the 3 groups. So treatment 1, the control group, got 4 plants. Treatment 2, the half dose group, got 4 plants, and the full dose group got 4 plants from store A. Then they did a similar thing with the 6 plants bought from store B, and they randomly assigned a third of each of those plants to the different treatment groups. So the control group got 2 plants from store B, and got 2 plants for the half dose group from store B, and treatment group 3 got 2 plants from store B. And then they were able to compare the juiciness and tastiness of the plants. Okay, and so we could see how treatment 1 works regardless of which store the tomatoes came from. Okay, now you may be wondering, well, how do they do the random assignment? Well, it's similar to random selection in that you just treat, like in this case, the 12 plants as a little population. You might number the plants 1 through 12 and put all the little numbers in a hat, mix them up, and then draw out 4 numbers. And the first 4 that are drawn out would go into group 1. Then you'd mix up the ones that are left and draw out 4 more, and those would go to group 2. And then the remaining 4 would go to group 3. And then you'd repeat the process, number the block B tomatoes plants 1 through 6. Get back to my slide here. 1 through 6, and then draw out the first 2. They would go into the control group. Draw out 2 more numbers. They would go into the half dose group, and then the remaining ones would go into the full dose group. In a retrospective or prospective study, subjects are sometimes paired because they are similar in ways not understudied. Matching the subjects in this way can reduce variability in much the same way as blocking. Even in experiments, if you use blocks of size 2, that's referred to as matching. So matching is just a specific case of blocking. Blocking is the same idea for experiments as stratifying as for sampling. Both methods group together subjects that are similar and randomize within those groups as a way to remove unwanted variation. We use blocks to reduce variability so we can see the effects of the factors. We're not usually interested in measuring the effects of the blocks themselves. We want to know how the factors work, and we just don't want the effects of the blocks to mask how the factors are working. When the levels of one factor are associated with levels of another factor, we say these 2 factors are confounded. When we have confounded factors, we cannot separate out the effects of one factor from the effects of the other factor. A lurking variable creates an association between 2 other variables that tempts us to think that one may cause the other. This can happen in regression analysis or an observational study. A lurking variable is usually thought of as a prior cause of both y and x that makes it appear that x may be causing y. Our randomization is an attempt to control a random assignment of subjects to treatments is our attempt to control lurking variables. Confounding can arise in an experiment when some other variable associated with the factor has an effect on the response variable. Since the experimenter assigns treatments at random to subjects rather than just observing them, a confounding variable can't be thought of as causing that assignment. A confounding variable then is associated in a non-causal way with a factor and affects the response. Because of the confounding, we we find that we can't tell whether any effect we see was caused by our factor or by the confounding factor or by both working together. Okay, so confounding variable, looking variable are both things that can cause problems. That's the big thing you need to know. Okay, go ahead and do the problems to try on your own, and I will see you in class. Thank you for taking good notes. I hope you have a good day.